Well, hello, AP Bio students. Mrs. Shelton here, and I am here to talk to you about Chapter 1. So if you are listening to this as a screencastify, then that means that you didn't listen to the lecture during Zoom today about Chapter 1 content. But what I want to make sure, since this is our first chapter that we're doing together, you understand the process of what notes are going to be. And I gave you a note here and directions to understand what I am providing you. So, what I am providing you for every chapter is I create an outline of the important concepts and terms from the chapter that you need to complete. So, there's a lot more that the textbook actually includes, but I need you guys to remember from your freshman biology what are prokaryotes and what are eukaryotes? Transcription and translation. So chapter 1 is going to be review, and I give you the notes outline for you to pull out the main content. And this is the content that will be testable. Now, I will always cover the content in lecture form. Your textbook covers the information too. There's other tutorial videos that cover it. But in your notes, please note please note in your notes, please understand that I view notes as being something personal. Notes are designed to help you summarize the information. Now, you could just take this Google Doc that I am providing for you, and you can just write your notes and write your explanations onto this Google Doc that once you turn it into a Google, your own copy for yourself, you could do that. If you want to print this out and then handwrite notes on it and then submit a picture to me, just insert a picture onto this document before you submit it. If you want to do flashcards, you can do that. Notes are personal. Notes are to help you summarize your understanding of the material. So if you look at a term and you're like, yeah, I already know what biology is, I know it's the study of life, you don't have to write it down if you don't want to. I'm not ever going to critique the quality of your notes until you're not being successful in the chapter tests. So you will have chapter tests that end up testing your knowledge. And that's where I'm going to go back and go, yeah, we need to step it up on these notes and make sure that you're not writing too much. Because some people end up writing way too much on their notes and not summarizing it and not understanding what they're writing. And some people just kind of skip over it and think, yeah, I got it. And then they really don't either. So again, notes are personal. Complete them in any format that you want or leave things blank. I'm not going to be looking and going, ooh, they didn't define what biotic and abiotic are. Well, guess what? You're going to need to know that on the test. And if you don't define it here on your notes, then it's going to show up on the test. So these are things to make sure that you are aware of when it comes to notes. Now, at the bottom of this document, though, this is the part that is not negotiable. The free response questions are questions that you have to answer that are pertaining to Chapter 1. When you answer these questions, you must write in complete sentences and you must write in your own words. This is practice for just free response writing for the College Board. That's half of your AP test is free response writing. But these are very chapter specific questions. Most of them require 1 or 2 sentences, so they're not long essays or anything like that. They're pretty much short, sweet, to the point is what they are. So that is it on the notes. Again, if you have questions, just check with me. But what I want to go over then is the content with you. So that way you are understanding the material. You've heard my verbal explanation before. Let's see if we can get this to show. This is not the way I want it to show up. So let's see what we can do. Hide presenter view. There we go. That's the way we want it. Okay. Maybe. Okay. So in chapter 1, it really is just review of biology concepts that you should have mastered your freshman year, but we need to make sure that we have some common language and common understanding as we go through chapter 1 information. So first of all, we need to recognize that biology is a study of life, and the core theme of biology is actually evolution, because what we see in all living things is that there's unity. So with that unity, that means that life has to come from previous life. Cells have to come from previous cells, and all living things are composed of cells. Therefore, there's this unity that exists of pre-existing organisms. Now, then there's also diversity as well. And so that diversity is also what evolution studies, is where has that change occurred in that accumulation of change that we can link back to DNA, link then back to protein structure and function as well, and we can start seeing this change. But all living things have cells, all living things have DNA, all living things actually then have metabolism, all living things have ribosomes. So there's this unity, but then we look at evolution as the diversity, and that really is what the study of biology is about. What are the similar biochemical processes and structures? What then are the differences as we diverged and life evolved on this planet? Now, something you're going to have to also recognize is levels of organization, all the different levels for which we can study biology. So, starting at the molecule level, so it can actually go down to the atom level as well. Molecules then form macromolecules, macromolecules then form organelles inside cells, those small little structures. This would be a chloroplast, would be one small structure. Organelles group together to form cells. This is actually the level in which life begins. Groups of similar cells then form tissues. Groups of two or more tissues form organs, and those organs group together to form an entire organism. So the organism is the full life form that has the ability to then reproduce. That those organisms of the same species then form the population when you now have multiple members of the same species living together, able to reproduce. Communities then is the combination of different populations in the area where there's interactions that occur. And then ecosystems involve all the communities, so all the living things, but then also the non-living things, so the biotic and then abiotic factors and how they react and interwork together. And then we've got the biosphere, which is all the regions of the earth where life can exist. So those are my levels of organization. You don't need to memorize them in order. However, you should recognize what a tissue is. You should recognize what an ecosystem is, the difference from a population to a community. You should understand that. And what you should also know too is that when we're studying biology, we always have to kind of break things down smaller and smaller and smaller. And that's what is called reductionism. So if we're looking at an ecosystem that's no longer thriving, well, we usually have to start looking down in this reductionism form in order to be able to kind of tell what might be happening. Maybe it's something at the molecule level where there's too much nitrogen, or the pH has changed in the soil, which now is changing then the plant life, which now is changing then the herbivore life. And so we always have to do a reductionism approach. Now, we also have to look, though, at what moving to larger levels of organization do too, and those bring out emergent properties. Emergent properties are these unexpected properties as we now make something more complex that we couldn't have predicted would have happened at the smaller level. So when molecules, let's actually just even take it down to the atom level. The atom level, if I put sodium, sodium has its own properties, chlorine has its own properties, we put it together, sodium by itself will kill you, chlorine by itself will kill you if you eat it, but now you put them together and you have sodium chloride. Well, now we put salt on our french fries, it doesn't kill us. Well, too many french fries and salt could kill you, but you got to do a lot of it. However, understand that it's a new emergent property as we move from atoms to molecules, as we move from molecules to organelles. There's new emergent properties that exist as we move up that are those unique properties that we couldn't have predicted when we were looking at it from that reduced, smaller form. In biology, it is very, very, very important that you know the difference between a eukaryotic cell versus a prokaryotic cell. So this is actually where two major domains of life diverge when we talk about the domain eukarya and the domain of archaea and bacteria. Prokaryote cells are single-celled organisms. They have a cell membrane, they have DNA, they have ribosomes, but these are our bacteria, is what prokaryote cells are, and they can only exist in a single-celled form. Now, prokaryotes lived on this planet for 1 billion years before any eukaryotes emerged. Eukaryote cells have organelles and membrane-bound organelles inside. The nucleus is encased in a membrane-bound organelle. They have mitochondria, chloroplast, if it's something that can perform photosynthesis, endoplasmic reticulum. So they have all of these structures, but they still all have cell membrane. DNA is a genetic material. They all have ribosomes for making protein. But other than that, when we now talk eukaryotes, they have organelles. And eukaryote cells, because of this compartmentalization, can be multicellular. We then are classified as eukaryotes. All of our cells are eukaryote cells. And that's when we talk about our different kingdoms within the domain eukarya. That's where we have kingdom Plantae, we have kingdom Fungi, we have kingdom Animalia, and we also have kingdom Protista. What we also have to understand, so as far as a theme, we have to recognize the importance of DNA and have some basics. We inherited our DNA from our parents, and our DNA then gets replicated, and through mitosis, every cell in our body ends up having an exact copy of the original DNA that we inherited. Now, that DNA contains genes. And so when we talk about DNA and genes, that DNA carries instructions for making proteins. A gene is a single instruction for making one protein, is the definition of that. And when our DNA gets expressed, some terms you should recognize from freshman biology is in order for that DNA to get turned into a protein, what first has to happen is transcription, where DNA gets copied into a messenger RNA sequence. And then after that, at the ribosome, messenger RNA has to get translated into that primary structure of a protein, that order of amino acids. So make sure you're comfortable with transcription and translation. And what we're referencing there is understanding how DNA codes for proteins. Now, in theme number 3, where life requires a transfer and transformation of energy, we're going to be talking a lot about photosynthesis and cellular respiration and lots of details. But in the basics, what I need you guys to understand, first of all, is that energy flows through living things, meaning that energy, ultimate energy source is from the sun, and then plants or any photosynthetic organism, is not just plants, but photosynthetic organisms will use the energy from the sun to then convert that into a stored chemical energy. That's what photosynthesis is. So photosynthesis is using the sun's energy in order to then transfer that energy into stored chemical energy of sugars and other macromolecules. And then through aerobic respiration, or even cellular respiration that's not aerobic, so anaerobic respiration, it's the breakdown and the using of that chemical energy in order to now do work. But then ultimately that energy gets lost in the form of heat, which is still, energy can't be created or destroyed. Heat is actually a very unusable, unorganized form of energy that living things cannot utilize. Now, as we're building molecules though, so as plants build sugar, they're taking smaller chemicals like carbon dioxide and water to convert those into sugar. And then through cellular respiration, we're breaking that sugar down and returning carbon dioxide and oxygen back to the soil and back to the atmosphere. And then that just recycles and recycles. So chemicals cycle, but energy flows through an ecosystem through photosynthesis and cellular respiration. Now, also what we address here is understanding that all living things interact with their environment. They interact with other populations and other species. And so terms we need to make sure that we're associating with this is the biotic factors that influence life in that area, which is all the living things interacting, and then also the abiotic factors. The abiotic factors are the non-living things, so the water salinity, the water temperature, the amount of nitrogen in the water. Those would be abiotic factors that are influencing all of the living things that are in this area. Here again, here's a lizard that is interacting with the environment. The rock would be an abiotic factor, the temperature would be an abiotic factor, and then the lizard would be the biotic factor, the living versus the non-living. Now, what we also have to make sure that we're addressing in biology is we understand just the basics of evolution. Evolution is that change over time of living things. And so what causes that change over time is that process of natural selection. So in the process of natural selection, we know that there's always an overproduction of offspring with heritable differences. And those heritable differences then lead to a differential reproductive survival rate and reproductive rate. And so this is what leads to adaptations, which are these characteristics that are better suited for a given environment. Now, through the process of evolution, when we start talking about macroevolution changes, well, we know that these 3 are all birds, but what has led to all of these differences is hundreds of thousands to millions of years of natural selection. and adaptations that have led to these differences within the birds. There is unity and there's also diversity that exists. Now, what we also, and this might be kind of new to you, I don't think freshman biology addressed this. Everything else should have been addressed in your freshman biology classes, but what we need to make sure that we understand is how life gets classified. And in classifying life, we need to understand the 3-domain system, and then within the domain eukarya, the 4 different kingdoms. Now, what a domain is the first way we classify life. And with all of the living things that currently exist on this planet and have existed on the planet, that's 3 billion years of life forms that we need to classify based on their evolutionary history, based on the unity and then the diversity in which they have acquired. Now, domain bacteria and domain archaea, these are 2 domains of prokaryote cells. The difference between these two is domain bacteria. This is your typical bacteria that you would expect to be on you and in you right now. And you actually have more bacteria in you and on you than you have your own cells. So there you go. There's your gross factoid of the day. Domain archaea, however, these particular bacteria, so these are prokaryotes, but archaea actually can live in extreme conditions. And so these would be ones that live in extreme heat. When you go and see like these boiling mud baths in Yosemite or up at Lassen National Park, and there's these bacteria that can live in high acidic environments or high extreme temperatures. These are the domain archaea that have way different biochemical processes that have branched off from domain bacteria. So they're in a different category because they have a lot more diversity from what used to be all lumped together. As when I was in high school, we didn't have 3 domains. When I was in high school, we actually had a 5-kingdom system, and all bacteria, all prokaryotes got put into what was called the kingdom Monera, is what that. But we now know through biochemistry that domain bacteria, domain archaea, way different, therefore they're classified completely differently. The way that they are connected, however, is they both have prokaryote cells. Now, all eukaryote cells are in the domain eukarya. Now, from that domain eukarya, we actually will break and classify organisms into then the next subgroup, which is called a kingdom. And so from kingdoms, we have the plant kingdom, we have the fungus kingdom, the animal kingdom, and the protist kingdom. Now, if you are a plant, you're not. You are obviously listening to me, so you're not a plant. But what plants have that's different from everything else is plants have a cell wall, and plants are autotrophs, meaning that they can do photosynthesis. That's what you can do. You are multicellular eukaryote with a cell wall that can do photosynthesis. You are now classified in the kingdom Plantae. Period. Now, there's other subdivisions that we'll talk about as we talk more about classification. Now, if you're in the kingdom fungi, well, you are multicellular eukaryote cells. You have cell walls, but you can't do photosynthesis. You're a heterotroph. And so you have to get your nutrients from surrounding decaying organic material. So that's the big difference between a plant versus a fungus is autotroph for plants can do photosynthesis, heterotroph if you're a fungi, but both have cell walls and both are multicellular eukaryotes. Now, kingdom Animalia. In kingdom Animalia, multicellular eukaryote. However, there's no cell walls on our cells, and we're also heterotrophs. We cannot do photosynthesis. We have to get our energy from alternative sources. And then last but not least is this dumping ground called the kingdom Protista. And the kingdom Protista, what you're going to probably see if you ever become a high school biology teacher, you'll go, oh, back when I was in high school, there used to be this kingdom Protista. This is currently the dumping ground for single-celled eukaryotes that we don't know where to put them yet because of biochemical classifications. Some of these can do photosynthesis, some of them can't. Some of them have cell walls, some of them don't. So this Protista group, we just say are the single-celled eukaryotes, and we leave them there. So that's how life gets classified, is based on their energy source and also cell structure is how they are classified. Now, in biology, what's really important is that we collect data. biology, science, it's all about observation. It really is just observation. And from that observation, we collect data that can be also observed by other people. We don't want to observe things that nobody else can. We want experiments that can be repeatable as well. And so data actually comes in two forms. Data that we collect can be qualitative data or quantitative data. And if I say quantitative, that means I can count it. It's a number, it's a value. But if it's now qualitative, it's descriptions. So both are important, but typically in science, what we really want to make sure that we are relying on is we are relying on that quantitative data that can be repeatable. Now, when we perform a controlled experiment, and this is a lot of our experiments that we do, they're controlled experiments where we're testing a hypothesis. A hypothesis is a testable statement. So if I made a statement that in a higher intensity light, the rate of photosynthesis increases, then I need to be able to test that in a controlled experiment. So it's a testable statement. Most hypotheses, kind of the easier form of a hypothesis, is it's got to be an if-then statement. If this happens, then this will occur. And you test it in a controlled experiment. Now, in a controlled experiments, and here's the basic rule in order to get your data, controlled experiments have everything the same. You have to have two setups for your, so in here, if I'm looking at this beach, I have to have two setups, and what I have to have is a control, and I have to have an experimental group. Everything has to be the same in the setups between the control and the experimental group, except for one variable. And here, the one variable in this experimental group is I've got non-camouflaged little rodents here. That's it. So in a controlled experiment, you can only have one variable that changes. Everything else needs to be the same, and your control group is what you use to compare it to. Now, when we talk about data, like in this graph, I've got dark and I've got light models, and then I've got numbers over here. Well, the light and the dark models, that is what is referred to as the independent variable. So this is the variable I chose as my experiment. I chose to have dark and I chose to have light. That's my independent variable. I chose it before the lab actually began. The dependent variable then is the results. The results of your lab is the dependent variable. Now in graphs, always independent variable goes on the x-axis, dependent variable always gets graphed on the y-axis. So that will cover in a little bit too. So again, when we do controlled experiments, what we always have to have is we have to have everything the same except for one variable that we are testing. So with this, full moon versus no moon. That's the difference. Everything is set up the same except for the dark coat and the light coat is what's happening here. And then I had my independent variable of the full moon versus the no moon. And then my dependent variable was then what I recorded during my experiment. So last but not least, if you're looking at this particular picture here, this is why biology is just cool. Living things are just amazing, especially when you start seeing some camouflage. So this is actually a gecko. Most of you would look at this and go, oh, it's just a scar on a tree. It's actually a gecko that has actually camouflaged itself there, which came to be through evolution. Now, when we talk about evolution through natural selection that actually led to the adaptation of this gecko blending into the tree, well, this is where we have to understand in science the term theory, because we talk about the theory of evolution. And please understand that in science, a theory is not a conjecture. A lot of people in regular common vernacular use the term theory just like we would use a hypothesis. It's just kind of our educated guess that's still not tested or proven. Please understand that theories are just very, very complex explanations for phenomena. They're ever changing. So where a law is also kind of on that even keel of a theory, but laws actually are going to explain things very simply. I can sum it up in one sentence and I can explain how something occurred with usually an equation if something's a law. A theory is way more complex. There's lots of other parts to it that now change the outcome and change the results. So just to give you an idea with a theory, theories being more complex, you guys, if you took chemistry your sophomore year, that entire course that you took in chemistry was atomic theory. there's a lot more that you haven't covered in atomic theory because there's so many moving parts. If you change the temperature, well, now it changes other molecules bonding and different reactions and pressure and temperature and all these things that now change. So there's lots of pieces to it that makes it very complex. So theories are true in science, theories are documented in science, theories are observed in science, but we can't just simply sum it up in one quick sentence. We cannot sum up how this gecko evolved in one quick sentence. There's 3 billion years of life and biotic factors and abiotic factors and mutations that have all accumulated in order to lead to this particular organism. So just kind of understand where theory, that term comes into in biology and when we're talking science. So with that, that is the end of the presentation. That is chapter 1 in a nutshell. Make sure you know those terms. This one more specifically than other chapters, it really is about memorizing the terms and making sure you're comfortable with those. So if you do have any questions, then come see me and